Okay, so what do you need to know about pancreas? Anatomically, nothing fancy. Posterior to the stomach. 3 parts which are not really distinguishable: head, body, and a tail. But 2 things. First, you have acinar cells. Okay, that produce bicarbonate, right? Because that's the big story about pancreas. And guess how they produce it? You can basically dictate it to me, I believe. So CO2 diffuses from the blood, tracks with water, right? Carbonic acid. Now this time you forget about hydrogen, and bicarbonate is what is being pumped into the ducts. Does that make sense?
So does the hydrogen just goes back?
Hydrogen goes back in the blood, pumped back in the blood. Okay? Makes sense, right? Now, that's bicarbonate component. Acinar cells also produce a lot of digestive enzymes. Okay? Now, I struggle with myself, like because I don't want to spend precious time on things that you can memorize, and I know you can. So there's a list of pancreatic enzymes. It's in the notes and it's in the study guide. It's like you just follow those questions, like what is the function of this? What is the function of that?
I think I remember that.
Yeah, but I want to highlight a couple of conceptual things. First of all, is that many pancreatic enzymes are produced as the precursors, like, trypsinogen, okay? And this is I want to outline. So trypsinogen is not an active enzyme. That makes sense? So that actually is brilliant because this enzyme easily breaks down proteins. So if you dump trypsinogen into a lumen of a duct in the pancreas, it will start digesting the pancreas. You see what I'm saying? You don't want it to be active until it enters the intestine. So when it gets into the intestine, it is activated by the special enzyme called enterokinase. We'll get back to this idea, enterokinase. So far, okay, let's put it this way. Remember that the intestinal columnar cell, the brush border. Enterokinase is in the brush border. So it is strictly in the intestine. When trypsinogen ends up in the intestine, enterokinase, that is in the intestine, activates it into trypsin. That makes sense? Now what about others? Well, just as an example, you have others like chymotrypsinogen. Trypsin activates it into the active form. Does that make sense? Same goes for proelastase to elastase, procarboxypeptidase to carboxypeptidase. Now, obvious question. What do I need and don't need to know? Because a lot of this stuff is totally Googleable. I want you to understand, basically like as a big idea, that a good chunk, basically all proteolytic enzymes that break down proteins, proteolytic enzymes from the pancreas get activated in the intestine. Trypsin gets activated first, right, by enterokinase. And then trypsin activates everything else. Does that make sense?
Yes.
And especially, I focus your attention on the fact that it's proteolytic enzyme and phospholipase. Now think about this, like this is genius. I'm not saying that it's design, it's evolution. Everybody had a different system, they did not survive. But think about it. Proteolytic enzymes, enzymes that break down proteins, can be absolutely detrimental to junction proteins, proteins that hold cells together. Actually, you can use trypsin to take tissue and break it apart into individual cells. It can be done in the lab. I did that myself. So you don't want proteolytic enzymes to be activated prematurely. Phospholipase breaks down phospholipids, so basically death to membranes. You don't want it to be activated prematurely either. So all proteolytic enzymes and phospholipase will be activated when they are in the intestine. Does that make sense? So know all the enzymes that are in the list. Enzymes that are produced by the pancreas can digest everything. Proteins, phospholipids, lipids, nucleic acids. Does that make sense? And now sort of a final big idea. Think about nutrients that we consume as a relatively big polymers, right? Like polysaccharide, like starch, glycogen. Proteins with hundreds of amino acids. So when you break them down, you don't break them to individual building blocks right away. Bring them into the chunks. So pancreatic enzymes break them into chunks. So you go from starch. So here's what you have. You have say you have a starch. Just as this is an example. I'm not asking to know this all small details, but starch, you break it into maltose, which is disaccharide, dextrose, which is like oligosaccharide, different, or maltose, which is trisaccharide. Does it make sense? And same goes for proteins. You break proteins down not in individual amino acids, but in short peptides. Because then you're going to have another set of enzymes that will finish the job. We're going to talk about them. But for now, if you have an idea, and again, what kind of question I can ask? What is the substrate of trypsin? What do you need? Protein. That's all. I don't need small details. What is the substrate of, I don't know, phospholipase? Well, it's kind of, self-explanatory, phospholipids. But that's a level of knowledge that I ask you to have here. Questions? No. Okay.